Hello, and welcome back to Honors Biology. So, we've just wrapped up talking about diffusion and facultative diffusion, two of the ways things can get into and outside the cell. Today, we're going to be talking about the third type of passive transport, ways things can get in and out of the cell, and that's osmosis. So, osmosis is a type of diffusion, but there's some little tricks with it. So in this one, just to rehash some of that vocab we talked about last time, right? Solution is two things dissolved together. The solvent is what's doing the dissolving, which is water. Solute is what is being dissolved. Sugar, salt, it can be glucose, it can be whatever. The reason why I bring this up again is because the solutes, this becomes important when we talk about osmosis. So the solute is the compound being dissolved. So make sure you recognize that as we kind of go through this. So let's talk about osmosis. Osmosis, what it is it's the movement of water. It's the diffusion of water through the cell membrane, through that selectively permeable membrane. So, and water will also move from high to low, same as diffusion. This becomes important and why we talk about just water. So water can freely move through the cell membrane, right? We talked about facultative diffusion, and we have those aquaporins in the cell that allow water to freely move back and forth. Why water moves is because the solutes, these green things, are too big for them to fit through the cell or even through a transport protein. They're too big. So instead, to help balance it out, instead of trying to figure out some way to make these green molecules move across, well, let's just take most of the water on the left side and just push it to the right side. So in the end, we end up with the same concentration across the cell membrane. So instead of having this be like a 5% concentration and this be like a 50% concentration, if we move water across it, it'll raise this concentration to, let's say, like 10%, and it'll lower this concentration to 10%. So that's why osmosis is the movement of water. Now, there's a couple of different ways that water can move in reference to the cell. When we talk about the cell, there's a couple different ways water can move. Water can move outside the cell. So if we place it in a solution and water rushes outside the cell, we call that hypertonic. If water rushes into the cell, we call that hypotonic. And then if there's an equal movement of water inside and outside the cell, it's isotonic. So let's break down what each of those words means. Hypertonic, hypotonic, isotonic. And we'll start with hypotonic. What a hypotonic solution is, it's a solution that has a lower concentration of solutes and a higher concentration of water than inside the cell. So if we were to place this cell here in this beaker of salt water, okay? If the beaker of salt water had 10% salt, well, inside the cell only has 20% salt. So the way to think about it is this inside the cell is a lower concentration of water inside the cell than outside the cell. So to balance out, water is going to move into the cell because salt's too big to pass back and forth between the cell membrane. So we're going to move water. So what this does is then water rushes into the cell. And eventually we can put so much water into the cell where the cell will burst open. So we see this a lot with red blood cells. We can literally get it to burst open. So hypotonic solution is when water will rush into the cell. The surrounding solution has a lower concentration of solutes than inside the cell, so water will rush in to balance it out. Now we can have the opposite. We can have a hypertonic solution. Hypertonic is the opposite of hypotonic. Hypertonic is where the solution, our beaker of water, has a lower concentration of water and a higher concentration of solutes than inside the cell. So our salt water here, as we see, right, the surrounding solution has a higher concentration of salt than inside the cell. So there's more salt and less water outside the cell than inside the cell. So to rectify that, water is going to move outside the cell. And eventually, we can get so much, and the cell's literally gonna shrink. So if we look at this picture, this is a normal plant cell that we see. Well, if it's in a hypertonic solution full of salt water, water rushes out and we can see the cell membrane separate from the cell wall and just shrink. And we can get it to shrink so much where it just kind of collapses in on itself. So that's a hypertonic solution. And then the third type of solution we have is just right. It's an isotonic solution. And this just means the concentration of solutes in the solution is equal to the concentration of solutes inside the cell. So we get an equal movement of water across both. So it's an equal movement in and out. This is what cells like to be at. This is that preferred area for the cell. So cell will remain the same size. It works great. And it's at equilibrium. So 3 types: hypertonic, hypotonic, isotonic. My recommendation is to make like a T-chart to keep them all straight. So perfect. Yeah, that's all I got. So if you have any questions, remember, write them down, ask me in class, and I will see you next time in class. Have a great day. Bye.